So by my clock, it's 2. So
Yes.
please take it away.
Yes.
Okay, so my name is Matthew Merritt. It's my pleasure to speak to you today on behalf of the Isotope Tracer faculty. I'm going to be talking about central metabolism as a target of Pioglitazone action. Elizabeth asked me to give a little bit of an introduction about myself. Actually fell in love with magnets when I was about 6, when my uncle took a fan apart and gave me the magnets, and I thought it was the coolest thing I'd ever seen in my life. And got to organic chemistry, and they took me in the room with the big magnets and all the electrical cords lying around, and I thought this is what I want to do. So my PhD is actually in physical chemistry, where I started out doing solid state NMR with Jake Schaefer at Washington University in St. Louis. I did another postdoc in solid state NMR at University of Washington. Then I broke my streak of being involved in Washington and ended up at UT Southwestern in Dallas, working with Craig Malloy and Dean Sherry, and came as the facility manager. And then in 2007, when we got our hyperpolarizer, I walked into Dean's office and I said, this is what I want to do. And that's truly the moment where I fell in love with metabolism. Now I think of myself as a metabolism person much more than an NMR person. So, but I'm really going to talk today with an MR set of lenses. I'm going to describe how you can use different methods to ask questions about metabolism. This is kind of a reason to talk about the isotope methods. We're really promoting the course, but this is work that I'm actually finishing up with Luke Norton, who's assistant professor at UT Health Sciences San Antonio. He was an attendee of our course, and while he was there, we just hit it off, and we've been working together since then, using tracers to understand some problems in hepatic metabolism. So I just want to point out that this is part 3 of the series that we've sponsored. Part 1 was Elizabeth Parks. I love this picture of Elizabeth. She's clasping her hands like she's nervous, but we all know that she's not nervous about anything. Then last time, Owen spoke and gave a great talk about glycogen metabolism and metabolic disease. So I've got quite the standard to live up to. So I'm just going to give you a little background on Pioglitazone. Right now, it's the only FDA-approved TZD for treatment of type 2 diabetes. Is generally acknowledged to be a PPAR gamma, PPAR alpha agonist. It is known that it increases expression of GLUT1 and GLUT4 in adipocytes. You can see how that might be a positive if you've got too much glucose in your bloodstream. It increases insulin receptor expression in adipocytes and hepatocytes. So this means that like the GLUT1 and GLUT4, this causes an increase in your peripheral glucose uptake. So it lowers your HbA1c, it lowers your free fatty acids through the insulin sensitization. All of this kind of works together. Also in that there's decreasing hepatic gluconeogenesis, but they do have some, the TZDs do have some side effects. For PO, you get weight gain. There is, despite the improvement in the metabolism, there is some association with increased bone fracture, so loss of bone mass, sodium retention, and edema. Most of these are these side effects are thought to originate through the PPAR targeting. So there's still interest in understanding how this drug works and elucidating the mechanism further. Now, pioglitazone also has other targets of action. It's implicated in reducing ER stress and ROS production. It is known to lower BCAA concentrations. So we know that they have a strong correlation with T2D as well. It's been shown to have beneficial effects in cancers. I don't know that literature well, but I can only assume that the increased quality of your glucose homeostasis probably has benefits in cancer. They can reduce cardiovascular risk markers, and they can also protect against renal injury. So I want to point out that there's been some recent work showing that pioglitazone can be effective for treating NAFLD and NASH. Ken Kuzi here at University of Florida published that paper in 2018 showing some resolution for NASH in patients. So this is a widely prescribed drug, and it could have extra benefits. And so it really is something that we want to understand backwards and forwards. Now, there are known mitochondrial effects of pioglitazone administration. Ken's lab, again, showed that in a mouse model with a trans fat diet of NAFLD, that the TCA cycle turnover was normalized in conjunction with lower inflammation on PO administration. And there's been reports of changes in pyruvate metabolism, primarily assumed to be through the mitochondrial pyruvate carriers. Luke did some of that work, and then also reference Kyle McComas and Brian Fink for their insights into MPC function inside the liver. So our hypothesis, calling all tracers, so I'm going to talk a lot about tracers. There's a great deal more to this research that I'm not going to show. This is with the idea of outlining what we can actually learn with deuterium and carbon-13 tracers in this case. But our general hypothesis is that insulin sensitization with PO ameliorates NAFLD by changing the hepatic pyruvate metabolism. And so we're going to go in and we're going to do our absolute best to fully trace what's happening to pyruvate in our model system. So we're going to be using BL6 and on the same background strain, OB mice. I'm going to primarily show you data from two groups. One is 12 to 14-week-old mice on this diet, and they were 4 weeks on diet, but some of them were treated with pioglitazone, so you'll see them denoted as OB control versus OB PO. And then also I'm going to show you a little bit of work in the perfused liver where we had an acute exposure with 1 millimolar circulating PO. All of this, of course, is IACUC approved. So just a little background information to frame the action of the pioglitazone. So on the left, you see estimates of the endogenous glucose production. We did this with tritiated glucose. And so you see that pioglitazone has the expected decrease in glucose production. Now, I'm going to make major use of that single measurement right there, because that is the quantitative, as in micromoles per minute, measure that we have. We can use that to index other measurements. So I just want to draw your attention to that measurement specifically. The fasting blood glucose improved. And then also using a clamp like what Owen described last time, we estimated we measured the insulin-stimulated glucose disposal, and you can see that these animals remain, the treated animals have increased glucose sensitivity, insulin sensitivity. Now, of course, if you turn around and ask about insulin resistance, it's the converse. So we make this measurement just by multiplying fasting insulin times the endogenous glucose production. So we see that our insulin sensitivity is just really, really activated. I mean, this is the type of change that you would want to see. Now, before I go any further, I'm going to be talking about gluconeogenesis and pyruvate utilization. Just want to stop. We measured lactate and pyruvate in the liver in the cytosol. As you know, that is a good estimate of the cytosolic NADH to NAD ratio. In this case, it was not changed. There wasn't even really much of a trend at all for a change in the lactate to pyruvate ratio. We need to keep this in mind because one of the primary ways that you can control gluconeogenesis is by changing that redox state. That is not the source of what we're going to be seeing here. Now, we're going to measure our sources of plasma glucose, where all that glucose is actually coming from, with a commonly used method called the D2O method that was first proposed by Dr. Landau at Case Western. And so this method, you just give, in humans, you can drink the D2O. In mice, you usually give an IP injection. And the deuterated water mixes freely into the body water. And the way that you can infer the sources of your glucose is just looking at this diagram. So the equilibrium between G6P and fructose 6-phosphate is very fast, and it's fast enough that you can assume that enrichment at this position matches that of body water, and that is very generally true. Now you can see that if your glucose is coming from glycogen, this is the only place that you're going to pick up a deuterium label. I should point out that there's plenty of other ways to label these other sites with deuterium, but their exchange, a lot of them are exchange reactions that don't allow you to make any quantitative assessment of the pathways. Now, the next deuterium that we're interested in actually comes on the H5 at the level of triphosphate isomerase. If your glucose is being, if it's gluconeogenesis from glycerol, then you will produce labeling at the H5 position. And if it comes all the way from the TCA cycle through PEP, then the H6S position can become deuterated from the body water, and that happens at the level of fumarase. So we can read out these three possible different sources of glucose by looking at the deuterium enrichment. And the way that Dr. Landau initially proposed doing this was going through this derivatization procedure, making the diacetone glucose, monoacetone glucose, but then ultimately it came down to a carbon by carbon degradation. This is a brilliant way to do this because once you get the CH or CD groups out from the carbon by carbon degradation and make this HMT derivative, you can see what happens is there's 6 different carbons inside this derivative. So that increases your fractional enrichment by a factor of 6. So that's a major sensitivity enhancement that can come by doing this. GCMS is already quite sensitive. So that means that this experiment, really you need a a surprisingly small amount of sample. The drawback of this approach is that you have to do a carbon by carbon degradation. So it is time consuming. Now, another possibility of a way to take advantage of this experiment is actually by magnetic resonance. Now, if you know anything about magnetic resonance versus GCMS, you're going, I would much rather use the highly sensitive GCMS, but there are reasons to use the MR. So this is the same diagram. These are the equations that tell you about the glucose coming from glycogenolysis, glycerol, or from the level of the TCA cycle. But if we just make that monoacetone glucose derivative, this is the type of deuterium in a Mars spectrum that you can readily achieve. Now, well, why do we want to make this derivative? Well, adding this isopropylamine group locks out the boat chair conformers of the glucose. That means you get half the number of resonances. Everything kind of gets jammed into its own distinct resonance. You'll notice that it's even numbered correctly, 1, 2, 3, 4, 5, 6 in order. And then to top it off, it's alphabetically filed. So this is a very nice derivative, and it lets you make these estimates of the sources of glucose just by looking at the relative area of these peaks. So you don't have to do any sort of carbon by carbon degradation to get at that. It's a single derivative with a single NMR spectrum. Now this is some of the data that we got from our samples. And so the way we did this was we gave 27 microliters per, let's see, what was that? Oh my gosh, I left out my experimental page. We gave a IP injection of D2O, and then we got the glucose out of the blood, did the derivatization, did these measurements, and this is the first place where we were really quite surprised. Our hepatic glucose output, the sources of endogenous glucose production were really not even trending toward very different, which really kind of took us aback. But the numbers were what they were. You can't argue with the spectra. But now I want to go back and point to the endogenous glucose production. And since this is in micromoles per minute, we can take these fractional contributions and turn them into actual quantitative contributions to the production of the glucose. Now, the other thing that you obviously want to ask if you're interested in glycogenolysis is, what does the liver glycogen look like? And in the OB control animal, you can see that there's a massive upregulation in glycogen storage. And then the PO treatment lowers the glycogen content significantly, but it doesn't come anywhere close to restoring it to the wild type. But using another tritiated glucose experiment, we found that glycogen synthesis is actually increased in OBPO versus the control. So this is inferring that there's some kind of glycogen cycling that's probably going on. So we need Owen. If you saw his talk last time, he is an expert in understanding glycogen cycling. This is certainly inferring that we've got some going on, but to really nail it down, we would need to do some extra experiments. But this tells you that PO does have some significant effects, not only on gluconeogenesis, but on how glycogen is stored in these animals. So I'm going to stop at various points and just summarize things as we go along. So our decreases in endogenous glucose production come without modulating the sources of the glucose. Lower glycogen storage on PO treatment. Now, I want to just talk in a more straightforward manner about the NMR versus the MS. Both of these techniques have their pluses and minuses. It would do well to weigh what you're trying to accomplish before you decided on a method, and unfortunately also find out if you have a magnet that's available, which is quite a bit more expensive than a GCMS. So magnetic resonance has the advantage of this chemical specificity, where we see the site-specific enrichment of the deuteriums. The sensitivity in the GCMS, it always wins. But there's certain advantages for MR that endure. And so I reference a new paper by Stan Deja, where he went through this in a very similar set of experiments and compared GCMS to MR. What MR has going for it is that you can actually measure very low fractional enrichments without having to worry about a background correction that's associated with the carbon-13. So that suggests that NMR can be very useful for human experiments or any kind of large mammal experiments because you usually cannot afford to achieve a very high level of enrichment inside of a human. And so that ability to look at very low fractional enrichments by MR, it can be a significant advantage, especially if you're going to take glucose out of the blood out of an adult. You can easily get plenty of glucose at very low fractional enrichments to do these experiments. Now, the other thing I want to point out is that you can use deuterium and carbon-13 quite easily together with MR because they're invisible to each other because of the frequency dependence of the detection. And that means that the experiments are quite straightforward. Now, there's been some absolutely brilliant MS work recently by Antonowicz, also by Jamie Young, looking at doing GCMS with a multiplicity of different derivatives that because of the high energy that you get from GCMS ionization, you can actually do positional enrichment analysis of carbon and deuterium. So some of the advantages of NMR are being chipped away at just by people that are thinking very carefully about the experiments. But it still remains that MR does have some advantages with low enrichments. Now I'm going to come back to the biology a little bit and talk about protein expression and activity. So a lot of the common mitochondrial proteins have increasing expression. So here you see COX-4 is up in the OB animal. It's somewhat normalized by the PO treatment, but this anion channel is really not changed. Now that's not really a mitochondria, that's an outer membrane protein. So what we're seeing here is that if you look at citrate synthase, IDH2, a lot of the proteins that are in the middle of the TCA cycle that we know are mitochondrial in nature are increased and don't really go back down on PO treatment. So this is suggesting that not only is there a change in energy handling, but there's a change in mitochondrial biogenesis. But I want to go over here to actual citrate synthase activity. So even though citrate synthase expression is maintained, the activity decreases significantly. on the PO treatment. So this is suggesting that our Krebs cycle turnover is actually increased, but then goes back down on PO treatment. This, incidentally, is really similar to the trans fat diet data that Sri Laxmi and Nishant Suni published. So now we want to get into the carbon-13 tracer experiments. We wanted to look at how the mitochondria and in the hepatocytes were handling the pyruvate that was coming in. The NMR-based isotoper analysis gives us these unequivocal carbon-13 fractional enrichments because we can get this site-specific enrichment. Now, I just want to give you an example of how this might work, just to orient you. So if we have a substrate that can make a label in acetyl-CoA, in this case, the filled white circles are assumed to be a carbon-13 label. If we have a 2-labeled acetyl-CoA and it goes into the TCA cycle, then that would label the 4th position as citrate, go down to alpha-ketoglutarate, and then it would, through exchange, produce a glutamate that's 4-labeled. We look at glutamate because NMR is sensitivity challenge, and glutamate is the highest abundance amino acid in tissue. And so that single C4 enrichment, if you look at the carbon-13 spectrum, is just going to give you a single line. Now, in the second turn of the cycle, we've actually produced labeled oxaloacetate that can condense with the labeled acetyl-CoA in the second turn of the cycle. And now when we look at the glutamate, we have 2 different isotopomers, one that's going to make a singlet at C2 and a singlet at C4. But in this case, we got 2 carbon-13s that are next to each other. That induces a coupling in the carbon-13 spectrum. And therefore, we can tell the difference between these 2 M+2 isotopomers just by inspection of the carbon-13 spectrum. So this is not data from this current study. I just wanted to point out what a carbon-13 spectrum can actually generate for you. So because of these chemical shifts, we can look at the C2 versus the C3 versus the C4 position of glutamate. And then you can see all of this fine structure at these resonances has to do with the abundance of the different carbon-13 isotopomers, where the carbon-13s are next to each other, causing this extra coupling that you can read out. So these homonuclear couplings provide direct estimates of your isotopomeric ratios. So we can leverage that to make models of metabolic turnover that we can use to interpret our data. I want to point out the data I'm getting ready to show was acquired using a special NMR probe built with high temperature superconducting technology that's available at the University of Florida. You can actually apply for a grant to use this technology within the National High Magnetic Field Laboratory. So this is something that you should consider if you're sensitivity challenged with the MR experiment. So to look at mitochondrial pyruvate utilization, we actually decided not to do what we normally do, which would be kind of a steady state isotoper analysis where we would give label until it came to isotopic equilibrium in the animal. This was just kind of a single time point experiment. This method was actually proposed by Insuk Jin in 2016. And in it, we're going to administer 2,3-C13 labeled pyruvate. And if that pyruvate is carboxylated, then it will produce these centrally labeled oxaloacetates. And after a single turn of the TCA cycle, it's going to make 2,3-label glutamate. If it's oxidized through pyruvate dehydrogenase, then it'll make 4,5-label glutamate. And so you can do a non-steady state analysis of PC versus PDH flux just by looking at the ratio of these two isotopomers. And that'll give you the PC to PDH ratio. Here's my experimental slide. So the data, you can actually do all of these at the same time. So the mice were given an IP injection with 27 microliters per gram of body weight of D2O, and that produced the deuterium spectra that you saw. 30 minutes later, there was an IP injection of the pyruvate tracer. And then after only 8 minutes, the livers were freeze clamped in situ and the blood collected. So the livers we extract and get glutamate spectra, and then the blood we made the MAG, and you can do carbon-13 NMR on the monoacetone glucose as well. So here we are, stunned again by the ratios that we see. Here is the isotopomer associated with pyruvate carboxylation in the carbon-13 spectrum that you see. This will come only from pyruvate oxidation. So you see singlet, that's natural abundance, C4 glutamate, and then the doublet 4,5 is actually isotopomer produced by pyruvate oxidation. Here's the singlet, so that's natural abundance C2 labeled glutamate, and then the D23 comes from our pyruvate tracer. So this glutamate is our readout of the mitochondrial TCA turnover. And even though it's glutamate is labeled by exchange with alpha-ketoglutarate, it's a faithful reproduction of the alpha-ketoglutarate labeling in most cases. So you can see here in OB control versus PO, there's just absolutely no change in the PC to PDH ratio. And in a clamp, it was not significantly different either. So I guess we shouldn't have been overly surprised by this. If the mitochondrial pyruvate carrier is really controlling what's happening, then that would suggest that we might expect this sort of result. But we really wanted to delve into this, make sure we weren't missing anything. So we can do the carbon-13 MR of the monoacetone glucose, not just the glutamate. So the blood is reading out the glucose isotopomers produced from this precursor. So we make these various oxaloacetate isotopomers on the first turn versus the second turn. They can go through PepCK and go up to glucose. You get this sort of labeling in the glucose on the second turn of the TCA cycle. And so we can inspect the carbon-13 spectrum and look for evidence of glucose production from the pyruvate. Now, this is, we talk about the NMR being insensitive, but this is actually pretty nice because after only 8 minutes, we can see glucose being produced from our tracer, and we know that it's coming from our tracer because it's this doublet. And you can see it in the C3 and the C4 position. And in the PO, you see that it's essentially invisible. If you've looked at this forever, you can see that that's a doublet right there. I could actually probably pick that out as a doublet. But that's really very poor enrichment. And so the way that you can interpret this data is either there's been a lot of scrambling, or you can reference back to the fact that the glucose production is actually much decreased in the PO treated. And so we just don't stand much of a chance of producing this because the total glucose production is actually quite a bit lower. Now, this is where I want to step back and talk about the strength of MR. So this singlet at the C3 and C4 position is actually natural abundance. That's 1% carbon-13. So because we use this doubly labeled tracer, when we see this doublet, we know that it came from the tracer, but you can also see that this natural abundance is 8, maybe 9 times larger than this doublet. So we can estimate a fractional enrichment by our tracer that's really way under the natural abundance. And so that is really the power of magnetic resonance. So the next phase is complete. Apparently, a PO treatment alters a PC and PDH flux concomitantly, but we really wanted to slam the door on that. And so we decided to also do some hyperpolarized carbon experiments. Now, I will not go into the physics of this. Basically, we're going to take our pyruvate. We're going to dope a stable free radical into it at a low concentration. And then this system, this is a magnet, and we actually take our sample, immerse it in liquid helium, and then we pump on it. This is a big vacuum pump. It reduces the temperature to around 1 Kelvin, which polarizes the electron close to unity. And then when we put in microwaves at the electron spin resonance frequency, it transfers polarization to the coupled carbon-13 nuclei. And so that unity polarization of electrons can result into 40 to 50% carbon-13 polarization. Normally it's 1 in a million. So we can produce enhancements of 40, 50, even 100,000 times of the carbon-13 signal. Then you have to melt the sample. But once it's melted, you have a metabolic imaging agent. And we can use that to study metabolism in the following way. We take a mouse liver, perfuse it through the portal vein, and actually put it inside the magnet. So this is an NMR tube that's around your liver. We perfuse it with a Krebs-Henselt buffer, and then we inject the hyperpolarized pyruvate by catheter into the space immediately above the liver and let the flow just pull the pyruvate through and watch it be metabolized. So we measure O2 consumptions to make sure that the liver is functioning properly. We use 1 millimolar Pioglitazone versus our control that has no drug. We injected 1 C13 pyruvate, and we did fed versus fasted versus acute PO in fed animals. And so the idea is, okay, this is acute as opposed to a chronic addition of Pioglitazone, but we wanted to see if there was anything that we could see regarding substrate handling. So this is the type of spectra that you can get from the hyperpolarization. This is the 1 C13 pyruvate. It goes up quite a bit further here. We've blown it up so that we can look and see. So here's lactate equilibration through a lactate dehydrogenase. Alanine aminotransferase is active. This is a hydrate of the pyruvate. And then if it goes through PDH, it'll make a carbon-13 labeled bicarbonate because of the CO2 elimination. Or if it goes through pyruvate carboxylase, then it will make malate and aspartate, these 4-carbon TCA cycle intermediates, or aspartates in fast exchange with oxaloacetate. So these metabolites come from PC flux. these metabolites come from PDH flux. We've got fasted, fed, and fed versus PO. And you see that there's just nothing that's different across these in any way. So we conclude that acute treatment with pioglitazone also does not modulate PC versus the PDH flux. Now, if we step back and we look at PDH expression and activity, you'll see that PDH expression is actually up in the OB control and the OBPO, but the PDH activity is significantly reduced. And it turns out that that is simply being mediated by the kinase. And so you can see that the mRNA of the PDK is going up with PO treatment. So we're really downregulating PDH flux. And concomitantly, we must be downregulating that PC flux. So we've got these changes so far. How can we draw this together to really estimate flux? This is how you would normally do it. Unfortunately, the wheels come off for me before I get to a final answer here. We may be able to double back and get to a final answer, but I can certainly propose how we think pioglitazone is working. So a key theme in your tracer studies is that you get a set of relative rates by whatever method that you're using. So here is sources of plasma glucose. Here's PC versus PDH flux. So all these are relative, but now we've got absolute endogenous glucose production from the tritiated glucose experiment. So normally what you can do is you can take all this together to turn it into an estimate of the absolute rates. So here's my take on the absolute rates, but there's some major assumptions I have to make to make this graph, unfortunately. Now, what I've got here labeled endogenous glucose production, this is pretty ironclad because we know the fractional rates, and we know that that we've got this decrease in endogenous glucose production. So in micromoles per minute, this is a very fair way to process this data. So that drop in endogenous glucose production brings down all of these contributions once you go to absolute rates. So now we have to start making assumptions because we're not in steady state. So I'm going to assume there's no PK flux. Warning, that's a huge assumption. The reason I can't assert anything about PK flux in this situation is because this tracer does not lend itself in a non-steady state manner to making the estimates of PK flux by looking at relative ratios of some of the peak heights. You could say, well, you can see it in lactate. You can't really see it in lactate if there's any PK flux with this tracer, because it will make a singly labeled lactate, which is hard to quantify. However, if we assume that there's no PK flux, then we can say that PC flux must be equal to PepCK flux. You know, if we're going to make glucose, it's because of pyruvate carboxylation. And so that's where we draw this PEP production equivalence to PC. Now, I should point out that PEP production is doubled versus glucose production because it takes two PEPs to make a single glucose. But if we make this assumption, we've got PC flux because of our relative rates of PC versus PDH. This is the PDH flux. Now, this is where the wheels really came off for me. We do not have a good estimate of substrate selection, unfortunately, because we're not in steady state. That is most unfortunate because now I can only assume that PDH flux is approximately 5% of total TCA cycle turnover. That's what it is in most of the literature for the liver really prefers to oxidize fatty acids. But if you make that assumption for these two cases, then the guesstimate that you get out is that PO actually lowers TCA cycle turnover by quite a bit, and this matches the estimates of Kalevala Poly using a different tracer. So this is not ironclad by any stretch of the imagination, but it's certainly suggestive. We want to double back and really get to the final answer on this. So in conclusion, pioglitazone does indeed change pyruvate handling, consistent with MPC targeting, because if you can't get it into the mitochondria, then you can't get pyruvate carboxylase flux, you can't get PDH flux. But it appears to be primarily in the context of decreased TCA cycle turnover. The reason I say that is that pyruvate availability should not be driving TCA cycle turnover in the liver. And so I think it takes a very nuanced view of turnover in the liver to be able to interpret this. But the upshot is that some PPAR sparing MPC antagonists could actually have other effects, because some new drugs that are not targeted at PPAR but only at MPC have shown some similar effects to pioglitazone. But I want to suggest that some of those effects are not primarily related only to the mitochondrial pyruvate carrier, but could be an off-target effect that's not understood as of yet. So in summary, just kind of an overview of the isotope methods, intermediary metabolism is really an ideal target for tracer studies because our atom mappings are ironclad. We know exactly where everything's going. And so, there's outstanding labs at Yale, Minnesota, in Nashville, at Vanderbilt, all over the place that know how to do these experiments. And if I didn't cite your paper, I really apologize. But this technology is so widely used that you can't keep track of it in a single talk. And I just want to put in a shot for NMR in that we are sensitively challenged, but we can do some really interesting things because of the technology. So finally, I want to acknowledge Dr. Raghavan, postdoc in my lab that's now associate faculty. If you need somebody that knows how to do, if you want to add a faculty member to your lab that knows how to do isotope tracers, you could do a lot worse than Dr. Raghavan. Luke and his postdoctoral associate Chris also as well from San Antonio. INSUC for helping us get the deuterium NMR spectra funding, and especially the R25 that supports our conference that we hopefully will have again one day. And so thank you very much. I will be glad to take questions.
Oh, we'll clap for you.
Thank you.
I guess for me, particularly with the use of a drug like Pio that may be affecting so many pathways at once. This technology looks like it's perfect for that. And I'm really wondering how close are we to understanding how this drug is working from the mouse studies, from the rodent studies?
Well, the work with the mitochondrial pyruvate carrier, I think, significantly enhanced our understanding that it's not just only PPAR associated. We're still learning things. You know, this final conclusion that I have here, you know, I don't think it's well understood the mechanism that how PO would downregulate TCA cycle turnover in such a significant way. You know, that's something that I think bears further looking into. That said, you know, we have learned a lot, but there's still, you know, if you're making a layer cake, there's still one more to put on top, in my humble opinion, for understanding the effects of this.
Thank you for that. Questions from the panel? Any?
No. Henry?
You have outlined some recent progresses both in the mass spec and in the NMR. And my question is, at this stage, can one measure gluconeogenesis with deuterated water at the recommended body water enrichment of 0.5% as Bernie Landau had recommended? Or are we not yet at that level?
But by the MR method?
Yes.
Yeah, definitely. So there's the advantage that you get is the amount of glucose that you can harvest from a blood sample from a human. And so even though you've got terribly low enrichment, you've got enough glucose that it becomes possible.
So you need to get what, 20 to 30 ml of blood for this?
Yeah,
So,
that's kind of the current level. I think that's likely to continue to drop
yes.
Due to improvements in MR probe technology, and so yeah, I'd say 20 is pretty easy at this point.
Now, for the mass spec technique with these multiple derivatives and propionate and so on, can you now go at 0.5% D2O?
So I would defer to Jamie Young and Stan Deja about that half a percent D2O with their methods. I have not used their methods personally. I think you can get there with, but if you get, you know, that's a case where your background correction for the carbon-13 and the silicons from that single derivative, you need some pretty sophisticated modeling to do it. I think it's possible using that Inca software that Jamie's developed, but you really need to have your head on straight. I wouldn't just sit down and start trying to do it one afternoon. You're going to get yourself in a lot of trouble.
Thank you.
Matt, it was nice seeing that work. One of the things you pointed out in the very beginning is that you measure oxygen consumption when you did the perfused livers, right?
Right.
As a way. So, and one of the interesting things is that basically the relative gluconeogenic flux to TCA cycle flux is almost constant. So basically every time gluconeogenesis is high, TCA cycle flux feeds that cycle. So what my question is sort of like, if you sort of step back, do you think it's the carbon availability that actually is driving the phenotype of the Pioglitazone liver rather than the intrinsic property of the TCA cycle and the gluconeogenic enzymes in this context?
That is a fascinating question because, as you know, it takes 6 ATPs to make a glucose. And so that ATP availability, if it's driven by the TCA cycle turnover, so that plays into that mass balance as well. That's the central point that I'm really coming around to. I think the pyruvate availability, you could say, okay, that's driving everything, but the pyruvate carboxylation at most can only generate 1 ATP as opposed to its oxidation. And so I really think that this data is pointing to the fact that there's something going on in the TCA cycle that is not just, I don't think shoving more pyruvate through pyruvate carboxylase is going to make the TCA cycle run faster or slower. I think there's something down there that's happening that's down-regulating the TCA cycle concomitantly with the decrease in the pyruvate availability.
And more of a technical issue, for most of your infusions of isotopes, say, for example, pyruvate, is it essentially viewed as a flooding dose approach? In other words, the number of moles you're giving relative to the endogenous moles of the animal is significant, right, when you're using NMR approaches?
Yeah. So that dose of pyruvate is indeed going to, massively change your circulating pyruvate levels, because that lactate we generally assume is 10-to-1 versus pyruvate. So there's no doubt that there is a little bit of an oxidative load associated with that pyruvate. And that's something that we just can't get around, unfortunately, as of yet, from an MR perspective. So we are kind of treading across the line of tracer sometimes with these experiments. Now, I should point out that is this specific approach that I'm using here. There's plenty of other approaches that I can point to where I think the tracer assumption is much more valid for MR. We just kind of hit it with a hammer here because we wanted to make sure we got an answer.
Can I add though that it's not that much different than becoming acutely fed. Not too far out of the amount of pyruvate that's in the liver. Maybe less, but it's like it's not too far off. It's not non-physiologic. It's just that the excess is labeled.
Okay.
Matt.
Yeah. Hey, Steve.
Hi, Steve. Thank you for the presentation, and also, for the other two before this. As you said, the whole putting all these three together is really useful. One of the things you comment on is obviously how to give the tracers and all these mixtures, and then how the NMR can sort out the individual labeling patterns. You kind of got at how the assumptions, when you then fit all these labeling patterns, can matter. Do we have a good review anywhere in the literature on all this, or is this something that someone like yourself, or maybe the guys at UT Southwestern who have all this simulation software, could sort of put together as like a kind of tutorial? Because as you said, one may think a pathway is not that active, 10%, 15% activity, and okay, we can ignore it. But that could actually have a big effect, right? When you're fitting all these models and trying to then make the interpretation out of it. And so is there something out there in literature, or is that something that, you, MMPC, should try to put together?
That could readily be reviewed again by the MMPC. I know that there was a big paper with Henry was one of the authors recently, Ralph Debradinas, Sean Burgess. It was I think over 20 different authors talking about isotope tracers and how they can be used for measuring metabolic flux. But I think reinforcing these concepts, of course, would be beneficial for everyone. And we should also probably try to engage, the metabolic flux analysis. I'm calling this isotoper analysis. I should point out that metabolic flux analysis is a little bit different flavor than this, and it might be useful for us actually to engage that community as well, and make a comment about how these two different ways of approaching the problem could actually synergize with each other, and how we could build a more robust product together. Because a lot of the MFA people, I think, generally have kind of an expanded vision. They want to use this any place they possibly can. I think about this a lot in the context of central metabolism, but there's no reason that we're stuck there. You just need a model that can describe where the label's going aside from that. Yeah.
Yep,
So
thank
Matt,
you, man.
just a general question, because you made a very important point in a lot of these models, is that you needed actually a foundational number called flux from which you can convert all the relative turnover rates to absolute. So if you had just given your deuterium and not had that glucose flux data, all you would know is that relative contributions changed or did not. But you would never have known that their absolute rates really did change. It's just their relative rates didn't change.
Yeah, and I mean, you're just hitting the point that I was really trying to drive home, that whenever you're doing these experiments, if you can find any place where you can get a quantitative estimate, you can use that as a ruler for everything else. And I have just noticed that over and over again, you saw that the fractional sources of glucose were not different, but when you scale them appropriately, you can tell those differences. And that happens over and over and over again. So I really encourage people that are thinking about using this technology, identify that kind of bottleneck reaction that you can use to turn everything into a quantitative rate, because it is much more likely to give you a result that you can interpret and help you understand your problem.
So the beauty you have in your perfused liver is you can measure how much glucose comes out of the liver. And that's your measure. And if you're doing a cell system, you could measure how much metabolite was consumed or produced in a period of time. And now you have a number that allows you to convert relative fluxes to absolute fluxes. And the other thing that's probably critical to what you were doing is that when you're studying TCA cycle, having some readout of what oxygen consumption is very useful, because it tells you what the metabolic demand is that TCA cycle is feeding. All right. So
that congruence between oxygen consumption and NADH production that comes off the TCA cycle can be so powerful. And so if you can measure oxygen consumption, measure it. It is very likely to tell you something very important. And the place where I feel like we've got room for growth is actually primarily in the heart. You know, a lot of heart failure situations, you can actually have a lot of oxygen wasting, which makes that oxygen consumption less useful. But you've got a bonus going for you in the heart, in that it's got a lot of ATP, so you can use the MR to measure ATP turnover. And so you can get back to those absolute rates by your ATP turnover, and you don't have to draw this coupled TCA cycle turnover with oxygen consumption necessarily from there. But that's definitely what's been found, is that it looks like you're consuming a great deal of material in a heart that's failing, but it's not giving you the ATP that you need to make it beat correctly. So.
Well, that's a good place to stop. And thank you so much, Matt, for this great description of this methodology and the findings. And we'll see you on YouTube. This will be posted by the end of the day.
It's my pleasure. Thank you very much, everyone.
Thank you. And thanks to all those who attended. Bye-bye.
Thank you.
Thank you, Ben.
Thanks, Owen.
Take care, Matt.